Most recently, he's been the director of the Center for Evolution and Cancer at the University of California, San Francisco, where he's been pursuing a bunch of different projects. There are many awards that you can see on his CV, those faculty who would like to take a look, including a new award from NCI with a substantial funding to pursue work on heterogeneity in cancers and how that can be used to predict progress. Today's topic, what elephants in evolution have to teach us about cancer. Carlo.
Great, thank you. It's a real pleasure. Actually, it's a real pleasure to be here. I appreciate the invitation and the chance to talk to you about this. And I'm going to tell you today about two stories, but I want to start with this intriguing observation. This is called Peto's paradox. Sir Richard Peto, in the 70s, observed that if every cell in your body has some chance of becoming cancerous, an elephant, which has about 100 times more cells than a human, should be getting proportionally a lot more cancer than humans. Right? And a mouse, which has 1,000 times fewer cells than a human and only a 2-year lifespan, shouldn't get any cancer. And yet, if you leave mice alone in a lab, about 50 to 90% of them will get cancer. Right? And elephants are not dying of cancer at the kind of rate you would expect in this. So there's something interesting here. In fact, there's been selection and evolution on large animals to suppress cancer better than humans. And that's the key here. Can we learn from elephants how to do better cancer prevention than we have currently in humans? So to answer that question, we actually have to take a step back and talk about the evolution of cancer itself. So 1 billion years ago, there were no multicellular organisms. If you think about that for a minute, there's no cancer. Right? Cancer is a problem of multicellularity. And so something happened between about 1 billion and 500 million years ago with the evolution of multicellular organisms. Now to get a multicellular organism, what you have to do is you have to get the somatic cells of the body to stop replicating out of control and start doing things for the good of the body in exchange for the germline cells to pass on the genes to the next generation. So there's a sort of covenant between the germline and the somatic cells, and you can think of cancer as the breaking of that covenant. It's the somatic cells that are starting to divide out of control, going off on their own. So this evolution doesn't stop with the evolution of, this evolution of cancer suppression doesn't stop with this transition from single cell to multicell. The larger the body you have, the longer you live, the better you have to suppress cancer. So these are the naked mole rats are up here because they live for 20 to 30 years, much longer than most rodents, and they don't seem to get cancer. There's been over 1,000 necropsies with no cancer detected yet, naked mole rat. But the same question applies to elephants and to whales. What is it that they have evolution has discovered that suppresses cancer? So there's this interesting process of the last 500 million years of the evolution of cancer suppression. We'd like to understand what evolution has done there. So that brings us back to Peto's paradox. And here's the statement of it that I already gave you. So we should think from first principles that cancer risk should scale with body size and lifespan, all else being equal. But all else is not equal because there are trade-offs in evolution typically, kinds of things like trade-offs between living longer versus having more offspring. And a mouse that's going to get picked off by a predator within a year or two, there's no selective pressure on that mouse to suppress cancer more than a year or two. Right? It's like that story of Henry Ford going to the junkyards and looking for the pieces of the cars that were not breaking down and relaxing the specifications on those pieces because you're putting in too much effort and energy and money on those things that are giving no advantage. So a mouse gains no advantage from cancer suppression more than a year or two, but an elephant and a whale have to suppress cancer over this very large scale, over long periods of time in order to live long enough to reproduce. Right? So there's been selection on that cancer suppression. So what has that led to? Well, the first thing to say is, the first question is, well, is this real? Do elephants actually get more cancer than humans? So maybe it does, maybe our first principles do apply. And this is data that Aaliyah Collin, one of my students who just got her PhD, did for her thesis. She extracted this data from zoo necropsies, and these are all mammals. And you can see the cancer rates on the y-axis here, body mass times lifespan on the x-axis here, and here elephants. Here's humans would be somewhere in here. They're actually not plotted because we're a special case. Actually, they would be up here. And you can see that cancer rates do not increase with body size and lifespan. And if anything, they decrease a little bit. All right, so Peto's paradox is real. Here's the actual data on elephants. So Aaliyah found a database of elephant necropsies, finding the cause of death. and found that overall there was about an average of 4% of elephants die of cancer. Now to contrast that, humans worldwide, it's about 13% mortality from cancer. In the developed world, it's more like 25%. And this is just broken down by age. So indeed, elephants not only are not getting more cancer than humans, they're getting less cancer than humans. They're doing much better at suppressing cancer. So what Leah found was that elephants actually have 20 copies of the most important tumor suppressor gene we know of, p53. Okay, and this is just the phylogeny of those copies. p53, if you don't know about it, is mutated in the majority of human cancers. But what's interesting about these copies is that one of them looks like a normal gene with introns. The other 19 have lost their introns. They look like retrotransposons. They've gone through an RNA stage where the introns have been spliced out, then been retrotransposed back into the genome and jumped around many times. And we can date this based on sequences of other closely related organisms. These copies appear to be about on the order of 50 million years old. So the next question, of course, is we usually think of genes that don't have introns as probably not functional. So that's the next question. It's intriguing that this was the only cancer gene of all the known cancer genes that has been so duplicated in an elephant. So this is the outlier. There's one other cancer gene in elephants that appears in 5 copies, that's LIF. Otherwise, they're at just 1 copy. So our collaborator Josh Schiffman in Utah took fresh elephant blood and human blood and exposed them to different levels of radiation and looked at the rates of apoptosis. These are at different time points, 1 hour, 5 hours, 18 hours, 24 hours. The white is apoptosis in elephants, the gray is apoptosis in humans. What you can see is that elephant cells are exquisitely sensitive to apoptosis and sensitive to radiation. So as soon as they get the DNA damage from radiation, they fall over. So there's one way of a multicellular body of clearing itself from possible mutations is just when you get DNA damage, don't try to keep replicating, just kill off that cell, clear it, you're fine. So we've got this intriguing functional assay as well as the genetics. Now we looked into whether these retro genes are being expressed, and here's a qPCR from that, showing that we are detecting RNA from these retro genes. These retro genes actually cluster into 2 groups. These are deletions in the middle of the gene of 10 over 5 base pairs. 10 or 5 codons. And so you can just put primers on either side of this and look at gene products and find both of these families of RNAs is being expressed, although at relatively low levels. We don't know yet whether they're expressing protein. Now the other intriguing part of the story is that people have engineered mice to have extra copies of p53, and when you do that and then expose them to carcinogens, the mice with the extra copies of p53 die less frequently than the wild-type mice, which get cancer. So these are 2 different kinds of carcinogen exposures, and the mice, these are called super p53 mice. The super p53 guys are much more cancer resistant. And I should say, going back to this theme of trade-offs, that when they first added p53 to extra copies of p53 to mice, what they found was that they were cancer resistant, but they aged quickly. The theory there was that the stem cell pools that were replenishing and rejuvenating the tissues were getting depleted because the stem cells were keeling over. They're super sensitive to any kind of damage, and you're draining the stem cell pool and making them age. What Serrano and his colleagues did here is they put the p53 gene under the endogenous promoter as opposed to an always-on promoter, which the initial work was done with the CMV promoter. Here under the endogenous promoter, p53 should only be turned on when it should be there, and these mice actually have a normal lifespan and are cancer resistant. So what about the whale? So we got funding to sequence the genome of the humpback whale. There had been no whale that had been sequenced yet, in order to pursue this question of Peto's paradox. And we haven't published the genome yet. It's still in draft form, but we have 97% of the coding regions done, so Leo was able to analyze the genes in the whale. And one thing she did is she looked for convergent evolution. So she took all the cancer genes, and the normal phylogeny should look like this. Elephant should pair with the hyrax, which is its closest related species you can see here. Whales should pair with dolphins. Chimps should pair with humans. And you can regenerate the phylogeny based on each cancer gene and look for this kind of case where the elephant and whale genes look similar to each other, abnormally similar to each other, so the evidence of convergent evolution. And when you do that, one gene dramatically pops out. So there's this gene UBE-D1, which is part of the ubiquitin complex, so genes for cleaning up the trash in the cells. In all the 30 mammals that have been sequenced, at the amino acid level, this is absolutely conserved, 100% conserved at the amino acid level. So all the mammals have the same UBE-D1 gene, except for elephants and whales. And elephants and whales, you can see up there, in the first alpha helix, have both changed the first 8 amino acids of their alpha helix to almost exactly the same sequence. There's one amino acid difference there you can see in the second position. Otherwise, they've both discovered the same UBE-D1 gene. So that's intriguing. I should mention that elephants also have 2 extra copies of 2 other tumor suppressor genes, BECN1 and TCEL7. So we're going to want to pursue those. The other thing I just wanted to tell you from Aaliyah's work is that she looked at the cancer genes across all the sequenced mammals, looked at large animals and long-lived animals, and this is the other thing that there was convergent evolution around, which was duplications of this tumor suppressor gene MAL. So horses have 8 copies of MAL. Microbats, the little brown bat here, has 2 copies of it. These bats can live up to 34 years, so they're not obviously large animals, but very long-lived ones. So that's the other lead from her work that I'd like to follow up on. And so I'll talk a little bit more about that tomorrow at the Chalk Talk. So what are the implications of this? Well, evolution is telling us that P53 is key and we should be thinking about how to protect it. It also shows that these large animals, because largeness has evolved independently, we didn't necessarily expect the same mechanism. In fact, what we're finding is different mechanisms of cancer suppression across on these different branches that have evolved large size. And in general, what we're doing here is really comparative oncology or comparative biology, trying to understand the nature of cancer suppression across life by looking at the different branches of life and seeing how evolution has solved the problem. So that's the Peto's paradox story, but cancer is intriguing to evolutionary biologists because it's a really dramatic example of multi-level selection. So there's selection on the organismal level to suppress cancer because obviously it kills us in many cases, but there's also selection at the cellular level to generate cancer, and I'm going to take you through that. So this is actually where most of our work has been done, is down at the cell level. So the most important thing to get about cancer is that it evolves among cells. So there's natural selection on cells in your body. We call this somatic evolution. In fact, natural selection only requires 3 conditions, which are both necessary and sufficient. There has to be variation in the population. That variation has to be heritable, and that variation has to affect fitness, that is, reproductive or survival. So among the cells in your body, you get mutations during your lifetime, so you get somatic genetic lesions, so you get variation among your cells. That variation obviously is heritable because it's encoded in changes in the DNA and also methylation patterns. And that variation does affect survival in some cases, so you can see suppression of apoptosis, for example, in cancer. In fact, if you look across the so-called hallmarks of cancer, these are the phenotypes that are common across virtually all cancers. You go down the list of the hallmarks people have identified. Each of these phenotypes, each of these behaviors of cancer are things that increase the survival or reproduction of the cells. I'm not going to take you through all of them. The only thing that doesn't obviously fit in here is that one of the major themes across cancers is that they're genetically unstable. That is, their mutation rate is very high. So the mutation rate facilitates this evolutionary process and generates these other hallmarks. But the mutation rate itself doesn't necessarily affect reproduction and survival. Although we can talk about that more. So the other thing to get about this whole process is it's not just a cell autonomous process of evolution happening within the cells. Of course, evolution and the fitness effect of any mutation depends on the context, the microenvironment in this case. So tumors are evolving within our bodies. They're being fed or not by blood vessels. They're being impacted by their immune cells and the fibroblasts around them. And so what you actually have, what's the field of microenvironment in cancer is actually ecology. So in fact, there's been very little work to take the theories and the understanding and analysis of ecology and translate it to cancer. And that's one thing that Athena Tippis really specializes on. Hopefully you'll be hearing from her at some point. So the cancers actually co-opt fibroblasts to send them proliferative signals. They co-opt endothelial cells to generate blood vessels called angiogenesis to feed the tumors. And there are various stresses and limiting resources. In fact, in general, we don't know what the limiting resources are for a tumor. So that's another area of ecology that we'd like to apply to cancer biology. And in case you're wondering, can there be enough time for evolution to happen within our bodies, here are some of the parameters of this. And you should know that there's a lot of uncertainty about all these estimations, but mutation rates, there's all different kinds of genetic changes, but they can be, for instance, copy number changes, deletions, amplifications happen on the order of 10 to the negative 6, 1 in a million per cell generation. We have generation times of stem cells, or particularly in cell culture, of 1 to 3 days. But the time to malignancy from the first mutation to the full-blown malignancy where you feel sick is somewhere between 30 and 60 years for most tumors, so on the order of decades. And an estimated number of generations of hundreds to tens of thousands of cell generations, and population sizes of these tumors between 1 billion and 1 trillion cells. And so you put these numbers together, and you can see that every cell generation, if you've got 1 billion cells turning over, and at every location in the genome has about a 1 in a million chance of mutating, in every cell generation, every point in the genome is being tested, it's being mutated, being changed. So it's testing all possible mutations, and so we should actually expect convergent evolution within a tumor, that some cell lineages will find the same mutations independently. In fact, that's what people are starting to see. Here's a bunch of references showing that we do have variation within neoplasms, and you can use any assay you want. Here's a really influential paper from Charlie Swanton, where they took multiple samples from a renal cancer and did exome sequencing, and then generated the phylogeny. So each of these tips here are a different region from the same tumor, as well as some of the metastases. And they found that SETD2 was mutated independently 3 times within this tumor. So one way I like to think about cancer is with these evolutionary diagrams that really come from Crow and Kimura in the 70s, where you have time on the x-axis and frequency in the population on the y-axis. And you can think about a point mutation that may be neutral, as having no effect on the reproduction or survival of the tumors. That clone carrying that mutation will just increase or decrease randomly. This is Brownian genetic drift. Sometimes you'll get a mutation that gives an advantage, like an activation of a tumor suppressor gene, and if that clone survives past the initial stochastic stage of low frequency, then it will tend to expand in the population. This is called a clonal expansion in cancer biology. It's called a selective sweep in evolutionary biology. So we expect a series of these selective sweeps. But the truth is we actually don't have very good data yet on the dynamics of progression at this evolutionary level. And one reason we don't have that is because most of cancer data is done this way. We take a single time point, we take a single sample, we grind it up, and we apply some assay to it. And when you do that, you lose all of the variation within the population, and you lose the dynamics of time, and you actually can't see evolution happening. And this is one reason why there hasn't been as much evolutionary biology and cancer biology as there might have been, because our assays have just not had the opportunity to see those changes. So I want to tell you one of the ways you can use evolutionary biology to improve cancer biology, and one of the major ones is around biomarkers. So most biomarkers in cancer biology is there a particular mutation or a particular phenotype in the tumor? So you take a sample, you measure it, for example, for the presence of HER2 new amplifications in breast cancer or estrogen receptor expression. So you're looking for the presence or absence of a mutation or the expression level. And this is difficult to do in an evolving system because you have variation across the tumor. So where you take your sample, you're going to get a different answer. You also have stochasticity in just what mutations are happening. So, and in fact, the recent attempts to resequence cancers show that there are very many sets of mutations that can produce a cancer. So if you're trying to do prediction based on a particular mutation, you may only be picking up the relevant, what's relevant to a small minority of your tumor type. And of course, there's just historical accidents that happen that structure what you'll find. And so when you try to do base biomarkers on the stochastic results of this evolutionary process, it's actually very difficult to make predictions. So instead, we've been thinking about how to measure the process itself. And I should mention here's this work from Charlie Swanton again. They took these multiple samples, they ran expression arrays on them to generate expression profiles. And previously people had identified these signatures of aggressive versus non-aggressive. And this is the these ones with red on this side and blue on this side look aggressive, and the ones that vice versa look not aggressive. And what we're pointing out here is that two samples from the same tumor would have given you completely opposite predictions about what this tumor is going to do. Right, so this is the spatial heterogeneity problem of where you take your samples. So we are trying to develop these what we call universal biomarkers, that is measuring the evolutionary process itself. And I say it's universal because all tumors that we know of, all types of cancer evolve. And so if you can develop, for instance, measuring the mutation rate itself, that should be a useful biomarker for measuring the speed of evolution across all different types of tumors. So that should predict which tumors are going to go on to become malignant, and it should predict which ones will typically evolve resistance easily. So there are four parameters that affect the rate of evolution. There's mutation rate, there's population size, and here we're talking about the population of stem cells, the cells that are going to be propagating the tumor. There's generation time, so the faster these stem cells are dividing, the faster that it will evolve. And then there's the selective effect of a mutation. How big of an advantage does a particular mutation give the clone? And you can measure this in terms of the rate of clonal expansion. So we would like to measure all of these. And when I initially started this work, there wasn't a good way of measuring mutation rate. And then later we found in the evolution of viruses literature that people had shown that if you take 2 time points and there's enough time between those time points to get mutations, to get evolution, that you can actually distinguish the mutation rate. So we applied that to this condition called Barrett's esophagus, which is a pre-malignant condition in the esophagus. And each of these rows here is a different patient, and we were able to measure the mutation rate when they were on NSAIDs versus off NSAIDs. NSAIDs are non-steroidal anti-inflammatory drugs that seem to prevent cancer in this condition. What we showed was that when people were on NSAIDs in general, they had an order of magnitude lower mutation rate than when they were off NSAIDs. An example of what you can do when you have these evolutionary tools at hand. So let me tell you a little bit more about Barrett's esophagus, because it's a wonderful system for studying this process. So it's associated with chronic reflux from your stomach, so heartburn. And in about 10% of people that have very bad heartburn and bad enough that they go into their doctors to get an endoscope, when you stick the endoscope down the esophagus, you can see this reddish area lining up just above the stomach. And that's an area where the normal multi-layered squamous cells of the esophagus have been replaced by a single layer of columnar cells. And so you can sort of see the blood through that single layer. Now, this is interesting for cancer research because having this condition of Barrett's esophagus predisposes you to esophageal adenocarcinoma with a relative risk of 30 to 100 times fold higher than if you didn't have the condition. So it sounds scary, but it's actually not that bad because less than 1% of people with this condition per year will go on to cancer. So it's not a very dangerous condition. It's just more dangerous than not having it. The other important clinical thing about this condition is that the only definitive way of treating Barrett's esophagus is to cut out that part of the esophagus, but an esophagectomy is an extremely dangerous procedure where the surgeons will kill between 8 to 23% of their patients. Okay. So you put that number together with that number, and you say it's not a good idea to take this condition out. Right? So instead of cutting it out, we watch it. We take serial biopsies. We take an endoscope down there. We take little biopsies of it looking for cancer. Now, if you find the cancer, then it's time to take out that part of the esophagus. But until then, we get to take all these biopsies and watch this process. In fact, the patients come back over time. So what you get then is a move from taking a single biopsy at a single time point to multiple biopsies at multiple time points, and then you can start watching the heterogeneity both over space and time and how these clones are changing and how they're competing. So that's how I first started working on applying evolutionary theory to neoplastic progression, this process of carcinogenesis. So it's a great human model of how this process plays out. As I was starting to say, when I first started doing this, we didn't have a way of measuring mutation rate, so the best thing we could do was measure how much diversity there is in the tumor at a given time point. And in fact, ecologists do this all the time. We go out into the field and we try to measure how much species diversity there is in a particular area of the field. And so what we were able to do is take the measures of species diversity from ecology and translate them into clonal diversity within the tumors. And we showed that if you measure at baseline the amount of diversity, and we took our cohort of over 250 Barrett's patients, we showed that the people that had higher diversity, the upper quartile, were much more likely to get cancer than the people that had low diversity in their tumors. The specific measure I'm plotting here is called genetic divergence. So it's when you take multiple samples and ask how much the genome is different between the samples. It's just a proportion there. It turns out that when you do this, you can ask, how do you define a clone, or what measures are you measuring a clone? And it turned out it actually didn't really matter. We could take the mutations and the lesions that are around the tumor suppressor genes that are driving clonal expansion, so they're selective, and define a clone based on that and measure diversity there. This is the p-value, 10 to the negative minus 4. Or you can take all the passenger mutations, the neutral mutations that aren't under selective pressure, and measure diversity there. And again, you get predictive power to see who's going to go on to get cancer. You can also choose whatever diversity measure you like from ecology. So divergence is on the upper plot here. You can also just count the number of clones. There it is, the predictive power there. You can use Simpson's index or Shannon's index, all the different measures of diversity we found that all of them were significant at the p less than 0.001 level in this cohort. So it seemed to be actually a robust phenomenon. The more diverse tumors are the more dangerous ones, at least in Barrett's esophagus. So then the question is, does it replicate? Because in medicine, we don't change medical practice based on a result in a single cohort. Right? Because there might be something special about that cohort. You might have just gotten unlucky and gotten a low p-value for random reasons rather than actually a signal. So we want to see that it works in a different cohort before we'd actually want to change how we're going to manage our patients. So we've teamed up with Sheila Krishnadath in Amsterdam, who has a completely independent cohort. They do something really interesting. They put a brush down the esophagus and they scrape cells off the surface. So now you're getting a sampling across the entire surface of the Barrett's esophagus at the single cell level. So the initial cohort I'm going to show you is actually 100 patients from the Mayo Clinic where Sheila initially worked. And we also have a set of, those are high-grade dysplasia patients, so they're sort of high-risk patients. Now Sheila has a low-risk cohort in Amsterdam, and we've got both cohorts now. So this is going to be a completely different assay. So single cells, we're going to do a FISH assay, so fluorescent in situ hybridization, and look at the copy number of particular loci. So for example, the P16 locus, the telomerase locus, the centromere chromosome 9, and the APC locus. And you can color these with different colors, and so you can look within a single cell, you can score 4 loci at a time. And she's got 3 different panels, so on different cells, but these 12 different loci. And so they've got an automated way of calling the spots and that are then curated by hand. And you get data like this. For cell number 1, there are 0 copies of P16 and 2 copies of the normal diploid number for the other loci. And you can see that all these cells are the same clone. They have the same genetics. These cells are the same clone. These cells are the same clone. And you can distinguish different clones and their frequency within the population this way. And here's what you get. These Kaplan-Meier curves are flipped upside down. These are normal now survival curves, but the same idea as the ones I was showing you before. So 3 different probe sets, calculating the Shannon diversity index on each of them, looking at the upper quartile, the more diverse tumors at baseline. These patients were more likely to go on to get cancer than the less diverse tumors. So independent cohort, independent assay, diversity predicts progression. So we're really excited about this getting to get a validation. It's not yet been published. That was with Shannon index. Here's the mean pairwise divergence, the measure of how different the cells are. Again, higher diversity is more dangerous, more likely to progress to cancer. So I said this is our universal biomarker project. So what we want to do now is ask, do these diversity measures translate to different tumors, not just Barrett's esophagus? In fact, what we started out doing, since I'm in a thoracic oncology division in the surgery department at the UCSF, we've got a ton of lung cancers. So we went to our tissue bank of lung cancers and we asked, if we take 2 samples from the same tumor, will the presence of the amount of diversity predict overall survival? So we've shifted now from looking at previously we're looking at a pre-malignant tissue and predicting who's going to go on to get cancer. Now we've got people with cancer, and we want to know who's going to survive therapy. And the theory here is that the more diverse tumors here will be more likely to have resistant clones in them, so whatever therapy you apply to them will be more likely to recur and kill the patient. That's the theory. So we did the simplest thing we could. We took 2 samples from the same tumor. We ran Snip arrays on each sample as well as a normal tissue. And then we looked at we calculated the amount of divergence, how different are those 2 samples based on loss of heterozygosity. So there's been either deletion or recombination to remove heterozygosity, which is usually a sign when it's selected that there's a tumor suppressor there. So here's the basic epidemiology of this cohort. It's 58 patients. It's mostly early stage tumors. That's because the late stage tumors don't get surgery. We say because you can't, once you have metastases, you can't cure the person through surgery. And a pretty even distribution across the different grades of tumors. Most of these people had been smokers, but a few non-smokers in there. Now, if you quantify the amount of divergence in each tumor and plot the histogram of it, you tend to get these sort of exponential distributions. There's a lot of tumors that have very low diversity, and then a few tumors that have outliers. And that's also typical in Barrett's esophagus. You tend to have these sort of exponential distributions. The mean genetic divergence was just, median was 3.3% of the genome differing between the two samples, but it went all the way up to 56% of the genome. And this measuring divergence is not just a measure of the overall messed upness of the tumor, the burden of mutations. There's a correlation between having lesions in the genome and having diversity within the genome. Here's an example of, these are two circus plots. The green is one sample, the blue is the other sample from the same tumor. The bars are showing loss of heterozygosity. So you can see on chromosomes 3 through 5, the whole chromosome has lost heterozygosity, but the two samples are virtually identical. So lots of lesions, but low diversity. Here's another patient where there were lots of lesions, but very high diversity, a lot of differences between the two. Okay. So we're not just measuring how many lesions are in the tumors. And here's the data. So the upper quartile people with more divergence, more diversity in their tumors were dying much faster than people that had less diversity in their tumors. P-value 0.008. In fact, genetic divergence was actually a better predictor than stage in this cohort for diversity. So this is a multivariate Cox model. So we're controlling for stage or controlling for divergence and seeing the effect of it. So I think I should pause there. I want to see if there's any questions about diversity things. I'm going to take you through one more result here before we finish up. Yeah.
Do you know anything about the dynamics of the divergence itself? Like, I would assume eventually cells, if they are too divergent, they eventually fall off on the kind of like some sort of error threshold or error catastrophe.
Right.
Sort of thing. So I was thinking, like, do you think that they will actually kind of like saturate at some point and then stay there?
Yeah. So this is an interesting theoretical question. There's this idea of an error catastrophe threshold. If your mutation rate is too high, then you can't maintain the adaptations in your genome. And the amount of information will basically get destroyed. And so you can't evolve if your mutation rate is too high. So there's some evidence across cancers, particularly in breast cancer. What they found was that the tumors that had the highest mutational burden actually had lower risk than the tumors that had moderate levels of burden. So there's actually a unimodal distribution. So low levels of burden and very high levels of burden were both relatively good profile, survival profile. But in the middle, when you had sort of moderate levels, there was the worst profile. We've actually been analyzing all the tumors in the Cancer Genome Project, the TCGA, American Cancer Genome Project, and finding in general across cancers that seems to be a pattern that's replicating, that the tumors with the most, particularly these chromosomal changes, tumors at the extremes have better survival than the tumors in the middle. A huge amount of lesions or very few lesions have better survival than just having a moderate level of lesions. So and the interpretation of that is this error catastrophe threshold. That's a long-winded way of distracting you from the fact that no, we don't know the dynamics of how that evolves. Yeah. Oh, sorry. I think they're recording, so posterity. Karen.
So your Barrett's model is focused on this metric of diversity, right? It's focused on somatic mutations. You don't really incorporate copy number variation certainly within that model because it's not really going to be there. But obviously,
germline copy number variation.
Somatic copy number variation.
Oh no, that's what it's based on. It's based on these Snip arrays.
From the Barrett model, or from this?
This is Oh yeah, so the Barrett model initially was with microsatellites, and now we've gone to SNP arrays. So I didn't show you data on the SNP
Got it.
arrays.
So you're incorporating also.
Yeah, yeah, so
Obviously, in different tumors, you're going to have different quantities of sort of copy number variations
yeah.
that might relatively apply. The second thing is, you talk about, single biopsies not being necessarily very predictive or prognostic, if you will, for outcome. And yet there are, as you know, marker panels that are very effective in terms of prognosis based on single biopsies and that have been validated in multiple large settings. And so
maybe let me not give you too extreme a position. So I didn't mean to say that single biopsies are not useful, just that they suffer from some of these sampling errors, but that doesn't mean that there isn't good information from single biopsy. In fact, that's a very nice way, and I'll come back to any remaining questions. It's a very nice way of leading into this next bit, which is how much information can you get from a single biopsy? So what we tried to do is, from this lung cancer data, is to ask if you look within a single biopsy, and if you had multiple clones from within that biopsy, and you looked across the genome, then if there's a clone that's covering about 20% of your biopsy, then you should see some mutations that all are showing up at about 20% frequency in the assay. If there's a clone that's 60% of the biopsy, you should see a bunch of mutations or lesions that are more around the 60% level. And that a sample biopsy with lots of little clones in it should have a lot of different allelic variation frequencies. So you can sort of quantify this is on the x-axis here is as the frequency of the lesion within the sample, and you can quantify the variance of that as a sort of proxy for how much within biopsy variations there is. And when we did that, and we took the the maximum of that variation between the two biopsies, then the people that had a biopsy with lots of variation within the biopsy were also more likely to die than the people that had more homogeneous biopsies. So there is good data you can get. Oh, this is the first hint that we have that you can infer some diversity data from a single biopsy that's also predictive. Now, was there another question that I missed before we go on? All right, well, we're almost done. Other people have taken sequence data and tried to develop scoring of estimating how much diversity is within that data, and that's again based on that if you look across the genome and you're finding mutations in one area of the genome that's at 10% of the reads in your sequencing and another mutation that's at different number, 20% or 40%, you can try to start estimating how much diversity is underlying all of that. They developed what's called a math score, and this is a head and neck cancer, and showed that more diversity within these exome sequences was predictive of overall survival. So genetic diversity is our first and most developed example of a universal biomarker. It predicts progression in Barrett's esophagus. It seems to predict overall survival in lung adenocarcinomas. It's better than stage in these early stage tumors, but of course we don't have a lot of variation in stage here. And actually we were able to show that you don't have to go through a lot of bioinformatic processing of these SNP arrays. You could actually base your assays, the analysis just off the single SNP data and still get good results. So this helps to remove some of the issues of the variation in algorithms of how you're going to do this, or the robustness of the algorithms. And the other nice thing about this is this can be tested in any cancer. We can get two samples, any spatially separated samples. And so we want to do that in a bunch of other cancers, and we've now been funded to try it in breast cancer. The other nice thing about measuring diversity is you don't have to work out all of the molecular biology of your particular cancer. You don't have to know what are the driver genes or what are the mutator genes. You can just quantify the amount of variation across the genome and get good estimates of some of the underlying evolutionary dynamics. And another thing to say about these diversity measures is they integrate all the different factors that generate diversity. So we can be you can be a smoker and be having a lot of mutations because you're smoking, or you could have a mismatch repair defect in your germline and getting mutations that way. You could have a lot of turnover in tissue, you have a lot of inflammation that might be generating it. All of it's going to eventually gets recorded in the genome, and you pick it up through these assays. And as I sort of alluded to, and actually showed you, you can measure diversity in all different kinds of assays and still seem to get predictive value. Now, there's some caveats here. The lung cancer cohort in particular is small. It's only 58 patients, so it'll have to be tested in an independent cohort before we can be sure that that's real. And we still don't know actually how far apart your samples have to be. We don't know this phylogeography of how much diversity varies as you move away in space. And that's going to probably be different in different tumors. So in conclusion, cancer is really a dramatic example of what evolutionary biologists love to talk about, this multi-level selection. So there's selection at the population level for, where cancer is killing organisms, so it's selection for cancer suppression there, but within our bodies, there's selection among cells for generating cancers. And these two forces are in opposition. The other thing to get here is that somatic evolution, or evolution, is the theory of cancer. And in fact, it is taught in medical schools as a standard way of understanding cancer. What's missing in medical schools is an application of the evolutionary tools and the implications of that for how to better understand and manage cancer. And so one of our major roles is to translate the tools from evolution and also ecology into cancer biology and show that we can do better in the clinic and in public health that way. I've shown you the start of our universal biomarker project, and also these comparative oncology results suggest what we should be targeting and doing to suppress cancer. So let me just put up my collaborators and grants, and thank you all.
Could I ask people to use the microphone? Carl, so it was interesting, particularly universal biomarker concept. I think that's an interesting idea. But keeping with your ecology analogy, let's say, it sort of reminds me as if you're out as an ecologist, you're counting the species, but you forgot to record the terrain and the climate you were in.
Yeah.
And so here, probably the driving force for all of this cancer is the immune system's response to the cancer itself. So it's the host response, largely through the immune system, that's probably driving a lot of the phenotypes you're seeing here and genotypes in the tumor. And so can you comment about how you can leave the other half of this whole system out like that?
Right. No, yeah. So, so I'm not intentionally leaving it out. In fact, the breast cancer grants we just got will be profiling the amount of microenvironmental diversity, including the inflammatory cells. And so we'll take multiple regions from the tumor, characterizing and quantifying the different parameters of the extracellular matrix and stromal cells and inflammatory cells in those different areas, and asking how different is the environment. Because really this mosaic of clones is overlaid on a patchwork of microenvironments. Is where you are near to a blood vessel, there's how much inflammatory cells there are, whether you're near a bone, whether you're impacted with a lot of collagen and desmoplasia. So indeed, there is, as I said, the ecology is underdeveloped here. And in fact, in Barrett's esophagus, the hints are that the inflammatory cells are critical, because what is the best cancer prevention in Barrett's esophagus is anti-inflammatory drugs. The kinds of lesions we see early on in the process are exactly the kind of lesions you expect from reactive oxygen species and inflammation. And in general, in cancer, there's chronic inflammation that's driving the whole process.
Okay,
So,
you should probably talk to us, because there's a lot simpler ways to do things than what you're proposing.
Okay, great, yeah.
So I want to follow up on that. So as we think about sort of the immune response to tumors, there's both the pathogenic inflammatory response, that may or may not have diversity embedded within it, but obviously there's the adaptive response, which is essentially and may mimic sort of predator-prey interactions. So you may actually see shrinking and alterations of diversity depending on efficacy of immune response. In fact, you may be able to capture some of that within some of the models that you've been doing.
Yeah, we would love to sort of analyze it within a predator-prey type context. And you can imagine that if there's a particular clone that's immunogenic, that clone might start shrinking and as it's getting picked off, and other clones might have an advantage, et cetera. And certainly we see, and you know this probably better than I do, all the immune evasion phenotypes that come up in cancer, that by the time it becomes a full-blown cancer, they found ways of getting around the immune system. So clearly the immune system has been a strong selective pressure that has driven the evolution of that evasion. Yeah, I think it's an important area to develop. Yeah. So, just time out.
Your
turn it over.
Oh, sorry. So in your diversity analysis, you showed two graphs. One is measured by drivers mutations, the other by passengers, and you show that they're equally good, which is sort of unexpected, isn't it?
Not necessarily. I mean, so the passenger mutations, by their name in evolution biology, they're called hitchhiker mutations. They're catching a ride with whatever's happening in the selective, driven by selection. And so you can measure what's happening in selection by looking at the passenger mutations. So you're really just quantifying how much diversity is there. And so essentially it looks like it doesn't matter where you look in the genome, you'll get that quantification. Now, if the neutral mutations or the passenger mutations should be a finer grained way of looking at diversity. That is, imagine a simple pediatric cancer that might only be driven by 2 mutations. And if you're only measuring the selective mutations, then there's only roughly 4 possible configurations you could see. And so you get a very crude way of measuring a clone. But if there's 10 or 20 driver mutations, then you're probably picking up most of the clonal structure. That's the sort of my intuition about what's going on.
Okay, so a different side of that question is if diversity is just a measurement of different types of cells, then diversity is just measuring the rate of cell proliferation. The higher the diversity, the faster the cell proliferate, which tends to make it as a more, say, malignant tendency to become cancer.
So there's a crucial step there. So diversity is measuring mutation, an interaction between mutation rate and selection. And I think you're connecting mutation rate with proliferation rate. Is that right?
Right.
Because you can imagine a highly stable genome, cells proliferating very rapidly and getting no mutations, and you wouldn't see that. But in general, yes, we think that every cell division is potentially mutagenic, and so there should be a correlation between proliferation rate and mutation rate. Yes. So a rapidly dividing tumor should accumulate diversity faster than a slow dividing tumor. And so yes, you may be measuring that. Yeah. Yeah.
So in your model, you don't take into account space, right?
So the Barrett's work, it relies on space to be able to pick up different clones and different biopsies, but we haven't really put those biopsies back onto space and asked, is this clone contiguous, etc.
So do you think that diversity would depend on where you sample in the tumor?
Probably, I think so. For example, some areas of the tumor are more diverse than other areas of the tumor. Yeah, probably. I mean, I think it's heterogeneous, and you wouldn't expect a well mixed system. So quite possibly, yeah. So the reality is you can't get very many biopsies in general, and so you have to just put up with that and hope that it comes out in a large enough number of patients and stuff that the patterns emerge. But ideally, what we'd be doing is sequencing every single cell from the tumor and knowing exactly which cell came from where and putting it back on the map. It's just not feasible at the moment.
So, Carlo, I'm interested in how early this predictive phenomenon occurs, especially in Barrett's esophagus, because Barrett's esophagus is a really odd, in fact, it's the only example in the body of a metaplasia going from squamous to glandular, and being associated with a glandular tumor then. And so I'm thinking that even when there's no dysplasia, which would put it into a tumor category, there would still be diversity in the Barrett's, in the metaplasia. Right. And most of the patients who are undergoing this incredibly labor-intensive and expensive surveillance of Barrett's don't need it at all and never progress.
Right.
So is diversity a better predictor of who's going to get dysplasia? And then once you get dysplasia, is that a better predictor than high and low grade by pathology for who's going to get cancer?
Right. So the Barrett's, the big cohort we use in Seattle, that's everybody. So only about a fourth of them have high grade dysplasia. So those results hold for the metaplasia.
So it's a better predictor than pathologic.
So we haven't published this, but yes, it's a better, I can't say whether it's a better predictor than dysplasia for progression to cancer. So in general, we haven't measured dysplasia as an outcome or as a predictor because there's so much variation between pathologists and how they call dysplasia.
That's why I'm interested.
Okay. Yeah, so one of the early things that Brian did was he got a bunch of Barrett's pathologists together in a room and got them to score dysplasia and showed that they couldn't do it reliably. So if you want to do your science...
And they can't do it at all if there's active inflammation. So that would be another reason.
It's a nightmare, yeah. So we tend to avoid that and just go for cancer as the outcome. There was a question back there.
I have a question about the connections between the first part of your talk and the last talk. So in the first part, you provided these interesting data suggesting that those species at the highest risk of cancer have a much larger number of copies of these cancer suppressor genes. And the last part of your talk, and maybe I didn't quite get this right, suggests that what you really don't want to get are cancers with a high diversity of cells or clones in them.
Yeah.
And so I'm just wondering, is there, looking at the first part then, is there any evidence, or are you pursuing the question of whether these suppressor genes can be fine-tuned in some ways that would not only reduce the risk of cancer, but also the risk of certain kinds of cancers that are particularly likely to have bad fitness consequences?
Oh, that's a really interesting question. So, right. So you can think about what evolution is trying to do in terms of cancer suppression. And one of the things it might be doing, that you're bringing out, is it might be trying to prevent diversity or somatic diversity. Yeah, so the P53 it's a genome protector gene, right? So reinforcing that does help lower the mutation rate or keep the mutation rate lower. And so presumably does also reduce diversity or helps prevent that diversity. The other thing we've been thinking about is that evolution doesn't really care about you getting tumors in terms of selective effect. It cares about getting lethal tumors, right? So this has been the implication of this has turned out to be a big problem for cancer screening. That is, there's been little selection on us to prevent benign tumors. And so, but when you go to screening, you find a bunch of benign tumors, and then the problem is over-diagnosis and over-treatment, and then how to distinguish which benign tumors are really dangerous and which aren't. So that's one thing we actually think about as well, is that most of selection at the organismal level has probably been on this transition from benign to malignancy, so preventing invasion and preventing metastasis, not necessarily preventing the growth of initial three-dimensional mass that doesn't usually really impact our health very much. Yeah, that's good questions.
So following up on Ron's question, it seems to me like the first part of your talk lends itself to thinking about imagining at least applications. Mm-hmm. But what does the second part of your talk say for clinical applications?
Oh, great. Yeah, thank you. So the universal biomarkers trying to measure the dynamics of evolution certainly has clinical applications for this risk stratification. In fact, this is exactly the screening issue. So you have these Barrett's esophagus patients, most of whom will never get cancer, and you should send them home and get them out of the system and not do these really expensive endoscopies every 18 months, right? So what you want to distinguish is the risky ones from the non-risky ones. And then if you have and this is true for most of the pre-malignant tumors, on average, roughly, it seems like 5 to 10% of them will go on to get cancer, but 90% of them won't. And so what we really need is this risk stratification to focus our resources, and also if there's any risk for our interventions, then you only want to do that in people that are at a high risk of getting cancer. You don't want to hurt all the other people that wouldn't die anyway. So that's one major clinical implication. But what I'm interested in particularly is once you can measure these evolutionary parameters like mutation rate, then you can look for drugs to slow the process of evolution as a cancer prevention. Right? So you can look for like these NSAIDs that seem to be lowering the mutation rate by an order of magnitude. But now that we have that measurement, can we do better than an aspirin for lowering mutation rate? Can you do drug screens to slow evolution as a way of doing cancer prevention? So that's a whole novel research program that we've initiated to slow evolution for cancer prevention. Yeah.
So I'd like to ask, have you done the same approach with primary versus metastases?
We haven't yet. That's one thing that actually Athena actipus is interested in. In particular, it's only metastatic cancer, as you know, metastatic cancer that kills us. And there's a hypothesis that metastases are evolving faster than the primary. Well, the question is, why is metastatic cancer so hard to control? That's the the central question here. And one hypothesis is that once you get metastases, they're in a lot of different microenvironments in your body, and so they're under different selective pressures that make diversifying. There may well be, one of Athena's hypotheses is that there may be be generations of metastases, so a micrometastasis that spins off secondary metastasis, that spins off tertiary metastasis, and you get selection on the ability to metastasize that could actually drive this whole process much more quickly. And then you get back to a metapopulation dynamic, which also sort of juices evolution. So we would love to study that. Clinically, it's hard to do. What you basically need is a rapid autopsy study so you can get multiple metastases from the body. But often by the time the person has died from cancer, the primary was resected long ago, and so just from a tissue banking perspective, particularly with something like prostate cancer, the primary was taken out 10 or 20 years before, it's hard to get all those samples. But people have started to take multiple samples of metastases. Well, I guess the other issue is people don't usually resect metastases, except for some special cases. So again, it's a tissue acquisition problem. But yeah, we would love to do that, but it's in the future. Yeah.
So would you expect to see this more in pediatric tumors versus late onset tumors after people are past breeding age and there's no real selection for those?
Would I expect to see what specifically?
This more. Would you expect to see more diversity in the pediatric tumors versus late
no?
onset?
So I actually expect to see less diversity in pediatric tumors. So for a couple reasons. So the main one just being amount of time. So you got to accumulate diversity, and these adult tumors that have been evolving for often 30, 40 years just have a lot of time to accumulate it. And the pediatric cancers only have a couple years. The other thing is that many pediatric tumors only require a few hits because either the child has inherited a bad copy of a tumor suppressor gene, and so again, there's less time necessary to generate the tumor that makes the child sick enough that we detect it in the clinic. So in general, pediatric cancers seem to be genetically more simple than adult cancers, and the people have made some good progress in understanding cancer by studying those simpler pediatric cancers. As I'm thinking about, for example, Mel Greaves, because it's not so noisy. But yeah.
So then the same application, though, if you have somebody who's past breeding age and they get cancer, there's not really a selective pressure on them.
Oh yeah, talk to the anthropologist. So actually, we confer a lot of benefits on our children after we pass reproductive age. So there's probably been a lot of selection to maintain our lifespan past reproductive age, because not only do we raise our children for 20 years, but then we also pass on social status and stuff that gives them more breeding opportunities and things. So there's actually, I think, good evidence that grandparents have selective effects on their grandchildren. Thank you.
Well, my question is, I thought it interesting that so many tumors seem to go through this kind of convergent evolutionary process. So the end-state tumor has a similar kind of mutational profile. Is it possible there's any other force at work besides convergent evolution? Could you have like a non-random mutation? Could you speculate along those lines?
Yeah. All right. So yeah, let me be clear and then speculate. So if you look across tumors and you do the genetics of them, there isn't much convergent evolution. That is, there's only a few tumor suppressor genes that show up consistently within any given tumor type, but then there seems to be a host of other genes that are probably selected, but as I sort of alluded to, there's many different ways of generating cancer. So there's little convergent evolution in that sort of view of it. But again, within any particular tumor, there is now emerging some evidence that you're hitting the same mutation multiple times. Mel Greaves' work in leukemia also showing that, and that these kids are getting, within the same tumor, they're getting the same mutation multiple times. And you see that when you try to generate a phylogeny and you can't do a nice branching tree because there's been convergent evolution. All right, so can I speculate about sort of other forces that would work here? It's certainly true that mutations do not happen uniformly across the genome. So there's some areas of the genome that seem to accumulate mutations more than others, and there's two reasons for that. One is there may have been uniform mutations, but there's been selection, so you only see the ones that are in one area. But clearly also there's areas of the genome that are just more fragile. So there's fragile sites, and we see those early on, or typically actually early hits in cancer. Yeah, so there's a really interesting hypothesis. Again, this is Athena actipicis hypothesis, that there may have been evolution to make parts of our genome susceptible to mutations, actually as an anti-pathogen adaptation. So this is what we call the Plan B hypothesis, and forgive me because this is a little bit science fiction-y. So the Plan B hypothesis goes this way. Pathogens typically have a much shorter generation time than hosts, so they evolve more quickly and they can adapt to whatever the host is doing. The exception, of course, is our immune system has this evolutionary dynamic that on roughly the same time scale as most pathogens. But imagine a pathogen, bacteria or a virus that does well, particularly well, when there's been a breach in our epithelium, a wound, right? So they're under selective pressure to keep the wound open. And the one thing that we sort of see in tumors often is it looks like a wound that's not healing. And so one of Athena's hypotheses here is that in some cases, tumors actually might be driven initially by pathogens that are trying to keep a wound open, and then the wound healing signals from extra proliferation just drive the cells to proliferate, et cetera. So the wound healing sequence is a very scripted thing in our genome, right? We have a way of doing that, and so pathogens should be able to evolve to exploit that, to hijack it, right, to hack it. So then the argument goes, well, maybe we have a Plan B in our bodies, which is when a wound is not healing, it means that we've been hacked, and we need to go to Plan B. And Plan B is somatic mutation. So start mutating our genome, and you get the right selective pressure. That is, any cells that can proliferate in that situation and close the wound are the ones you want. And so maybe some areas of the genome have been left more susceptible to somatic mutations as a wound closing adaptation in the presence of pathogen. So there's multiple steps of assumptions there, or hypotheses there, I should say. This pathogen hacking our systems, the Plan B, et cetera, but that would be another intriguing ways in which there would be non-uniformity. So I'm starting to give Athena's talks, I think. Yeah. She's a great theoretical biologist. There's a lot of really interesting ideas. Yeah.
So in your genetic diversity measures that you do you account for massive genetic catastrophes, basically, like recombination of some sort? Because you mentioned you use SNP arrays, and the easiest way to get massive genetic alteration is when you have basically just fuse two chromosomes together or some and creating such sort of genetic diversity.
Yeah,
and in many cases, there have been reports where it's some massive event that actually triggers the really malignant cancer instead of having point mutations accumulating, which will not generate the amount of mutation that you need to get cancer.
Right, so there's this observation also of chromothripsis, which looks like a shattering of the genome, or at least parts of the genome. Yeah, so it looks like Barrett's esophagus may have this as well. So when we do the SNP arrays, which I didn't show you this data because it's mainly done in Brian Reed's lab, we do the SNP arrays on our cohort, multiple biopsies, multiple SNP arrays, multiple time points. What we see is the progressors typically have a clone that has a massively messed up genome, like 20% of the genome is messed up. And it suddenly appears in one biopsy and then may propagate to a couple other biopsies over time. So we've started to entertain this hypothesis of what evolutionary biologists would call punctuated equilibrium, that is a sudden change, or what Darwin would call macromutation, or a hopeful monster is the term that I like from the 19th century. So these hopeful monsters tend to show up. Now there's some caveats to that observation. We can only see things at the biopsy level at the moment, so it's possible that these hopeful monsters actually slowly accumulated these mutations over time in a single crypt, and then the last hit allowed them to get out of that crypt and expand to the point that we could detect them. So it might have been gradual, but appear punctuated just because of our assay. So we actually now look like we're going to be funded to go down to the single crypt level and measure the lesions at single crypts to determine whether there's this sudden appearance of these clones or not. It's a good question.
More questions?
All right.
Okay, there's another chalk talk tomorrow at 10 o'clock, this very room. Welcome to join us then. Really well. Thanks very much, Carl.
Okay, thank you.